Hello and welcome to this video on brain anatomy and brain localisation. I am Tanya Chamberlain from the Division of Anatomy here at the University of Leeds, and it is my job to help you understand the basics of neuroanatomy throughout your programme. So what's coming up today? Well, we're going to have a closer look at the brainstem and introduce you to the cranial nerves, the majority of which come from the brainstem. We are also going to look at the cortex in closer detail. Specifically, the primary and association areas of the motor and sensory cortices, and how damage to these areas can affect a patient. So let's have a look at the learning objectives for today. After this video, and any further self-directed study, you should be able to locate and define the functions and key features of an adult brain. Distinguish between primary projection areas of the cortex and association cortex. Name and locate the primary and secondary motor areas of the cortex. Name and locate key primary sensory and secondary sensory projection areas. So now let's take a closer look at the adult brain and the primary and association areas of cortex. The first thing we shall take a look at is the brainstem, which I have outlined in pink. We are currently viewing this from a lateral perspective, but let's add an image from the ventral aspect and the dorsal aspect. This image on the left is showing the underside of the brain as if we were facing the patient, whereas the image on the right is showing us the dorsal aspect, or how we would view the brainstem of someone if we were to stand behind them. On this dorsal image, it is clear that we have cut away some of the important regions. Here is where the brainstem connects to the cerebellum, or the little brain. These cerebellar peduncles are white matter axons or tracks that form circuits between the cerebellum and the rest of the brain. The brainstem is made up of three parts: the midbrain, the pons, and the medulla, which is continuous with the spinal cord. The brainstem is important because it is where 10 of the 12 cranial nerves begin their journey. But what are the cranial nerves? Well, in short, the cranial nerves are a set of 12 paired nerves that come directly from the brain. There are 24 in total, but as they come in pairs, we only need to remember 12 names, and thankfully their names are fairly easy to interpret what their role is. They are numbered in Roman numerals, or identified by their name. The first cranial nerve is the olfactory nerve. The word olfaction means to smell, so the olfactory nerves carry sensory information to the cortex about smell. These olfactory bulbs sit on top of the cribriform plate of the ethmoid bone, where they synapse chemical receptors in the nasal cavity and transmit the impulse via the olfactory tract to the brain. The second cranial nerve is the optic nerve. These nerves detect images from the retina and carry the information to the visual cortex in the brain, where these images are interpreted. So these fibres are also special sensory or afferent fibres. Cranial nerve 1 and 2 do not come from the brainstem, but the next 10 do. Cranial nerve 3 is the oculomotor nerve. If we pull the word apart, it will help us understand its role. Oculo, meaning eye, and motor, well, that speaks for itself. The eyeball has muscles surrounding it that we use when we are looking around without moving our head. Most of these muscles are controlled by the oculomotor nerve, except for a couple that we will look at in a minute. So the oculomotor nerve contains efferent or motor fibres to the muscles of the eyeball. The next cranial nerve is cranial nerve number 4, the trochlear nerve. So, remember how we said the oculomotor nerve controls the muscles of the eye? Well, the trochlear nerve controls one of the muscles of the eye that the oculomotor doesn't. so this also contains efferent fibres. Cranial nerve number 5 is the trigeminal nerve. This hugely significant nerve has multiple roles. It carries afferent or sensory fibres from the 3 regions of the face via its ophthalmic, maxillary and mandibular branches. It also transmits general sensory information from the anterior 2/3 of the tongue. But it also innervates the muscles of mastication, so it contains efferent fibres too. The trigeminal ganglion can be seen here, but we have cut it away on this side. The trigeminal ganglion houses the cell bodies of the afferent fibres. Cranial nerve number 6 is the abducens nerve. Going back to the eye motor nerves, the oculomotor and the trochlear, the abducens innervates the remaining eye muscle, the lateral rectus muscle. This nerve therefore contains motor or efferent fibres. Cranial nerve number 7 is the facial nerve. Predominantly, the facial nerve is used for the muscles of facial expression, but we also know that the facial nerve transmits taste sensation from the anterior 2/3 of the tongue, and also general sensory information from a small patch of skin behind the ear. Therefore, the facial nerve carries motor, sensory, and special sensory fibres to and from the brain. Cranial nerve number 8 is the vestibulocochlear nerve. This comes in 2 parts. The vestibular part is concerned with the part of the auditory apparatus that conveys balance, whilst the cochlear part transmits information from the auditory apparatus that transmits sound. So the vestibulocochlear nerve contains special sensory fibres. The glossopharyngeal nerve is cranial nerve number 9. It provides special sensory fibres from the posterior 1/3 of the tongue for detecting taste. It innervates the parotid gland via parasympathetic fibres. General sensory fibres come from the oropharynx and the posterior 1/3 of the tongue, and motor innervation to the stylopharyngeus muscle of the pharynx. The vagus nerve is cranial nerve number 10. This super important nerve has multiple roles, from controlling the heartbeat to coordinating swallowing, phonation, and the gastrointestinal tract. This nerve carries parasympathetic fibres, motor, and sensory fibres. The penultimate cranial nerve is accessory nerve 11. This nerve provides efferent fibres to a small number of muscles in the neck and across the shoulders. And the final cranial nerve is the hypoglossal. These motor fibres innervate the intrinsic and extrinsic muscles of the tongue, with exception of palatoglossus. So that was a whistle-stop tour of the cranial nerves. So let's have a closer look at the rest of the brainstem. This is a dorsal view of the brainstem, and we have a few important structures to highlight. So that you can see them a little more clearly, I have sketched on some bits of interest. These bumps at the top of the midbrain are called the superior colliculi. They contain some of the nuclei responsible for processing visual stimulus. The inferior colliculi sit right beneath these and contain some of the nuclei for auditory or hearing pathways. Beyond that, we can see the trochlear nerve or cranial nerve number 4. These pass out of the brainstem and wrap around to the front, to the ventral aspect. The pons can be seen here, with the cerebellar peduncles. These attach to the cerebellum. Then we have 2 parts of the medulla, the open or rostral medulla, and the closed or dorsal medulla. We will be readdressing the brainstem in future videos, so for now let's have a quick look at the cross-sections of what we expect to find at various levels. This slice has been taken from the midbrain. We can see the oculomotor nerves emerging from their nucleus. On the subject of nuclei, I should briefly state that the nuclei within the brain are not the same as what we think of when we think of cellular nuclei. In neuroanatomy, a nucleus, or plural form nuclei, is a cluster of neurons in the central nervous system located deep within the cerebral hemispheres and the brainstem. The neurons in one nucleus usually have a roughly similar connections and functions. We can also see these red nuclei. That's their actual name. These are nuclei involved in motor coordination. We can see these white matter tracks here that are called the cerebral peduncles. These connect the cerebrum to the brainstem. This black part here is the substantia nigra, which is where the areas that modulate motor function are, and in which we find display pathologies in tremor disorders like Parkinson's disease. The next cross-section is midway through the pons. Dorsally, we can see the aqueduct, which is something we will discuss when we discuss the ventricles and the CSF. We can also see the trochlear nerve, a few more nuclei, and a few more white matter tracks. This is a slice of the rostral medulla. We are now able to see a lot of cranial nerves and nuclei that they emerge from. On the final slide, we can see one of the vagus nuclei, with one of the vagus nerves emerging from each side. And here we can see from a lateral view the sections that we have just looked at. The diencephalon is the bridging brain. It is what connects the faraway brain, the telencephalon, to the brainstem. The diencephalon is part of the cerebrum, and in this blown up image we are able to see parts of the diencephalon of interest. The first one is the thalamus, which acts as a relay station between different regions of the brain and the spinal cord. The thalamus acts a bit like a switchboard, directing messages to the appropriate area. The hypothalamus, as its name suggests, is somewhat below the thalamus, and this is the area of the brain that controls and restores homeostasis. It has neuroendocrine functions, detecting when there are too many salts in our blood indicating dehydration, or our core temperature. It releases growth hormones, regulates sex hormones, controls our thyroid and therefore metabolism. With regards to controlling our endocrine system or hormone producing system, there isn't much that the hypothalamus isn't involved in one way or another. The epithalamus, labelled here, controls and regulates our sleep-wake cycles by this tiny pea-shaped gland called the pineal gland. And lastly, although not visible on this image, we have the subthalamus. This has a role in motor control. So let's have a look at the meat and potatoes of the area for the bulk of what we need to cover for today. This is the telencephalon, the most advanced and highly developed area of the brain that sets us apart from the other mammals. The telencephalon can be seen on this coronal image here. Remember, these squiggles and wiggles have names. The rounded areas are called gyri, and the grooves are called sulci. The outer ring of grey matter is called the cerebral cortex. This is the region where we find neuron cell bodies and billions of synapses. Areas like the thalamus are also grey matter bodies, but these are not cerebral cortex. The name cerebral cortex is reserved for the grey matter crust of gyri and sulci. Other grey matter areas of the brain are collectively termed the subcortical grey matter, and are not the focus of this video. The white matter tracts are made up of myelinated nerve axons, and these can be seen travelling in a predictable way throughout the telencephalon. So the cerebral cortex has a series of subdivisions. We have primary projection areas. In terms of sensory pathways, this is where specific sensory pathways terminate. In terms of motor pathways, most of the major descending pathways originate here. We also have what we call association areas. Association areas are essentially the rest of the cerebral hemisphere, and these assist or interpret motor or sensory information. We also have a third subdivision in something called the lateralisation of function. What this means is that dominance of some functions occurs in each particular cerebral hemisphere. For instance, something which we will come on today is the dominance of the left hemisphere in relation to speech and language. Here we have the brain in all its glory, and let's recap the lobes. We have the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. But we also have some structures to identify in order to be able to talk about the projection areas. There are specific names allocated to each gyri and sulci. We don't need to remember them all, but there are a select few that we do need to know. These are the sulci, which we will find very useful today. We have the lateral fissure, which is a giant groove that runs deep into the brain and separates the temporal lobe from the frontal and parietal lobe. Then we have the central sulcus, which isn't as easy to spot as the lateral fissure, but it is still fairly identifiable. This is because it is a sulcus and not a fissure. A sulcus is more superficial in depth and therefore doesn't run as deep. We can also see that it runs down the medial walls of the cortex within the sagittal fissure and terminating at this gyrus here called the cingulate gyrus. The sulcus separating the parietal and occipital lobe is the parieto-occipital sulcus. Now we have named a few of the sulci, we are in a position to look at the primary projection areas of the brain. And the first up is the primary motor area. It sits rostral to the central sulcus, so this gyrus is simply termed the pre-central gyrus. The primary motor area is dedicated to every movement that we have learned to do, so every motor skill developed. This is where those efferent fibres originate before coursing to their destination. Located behind the central sulcus is the primary somatosensory area of the brain. Due to its location, this is called the post-central gyrus. It makes sense that the primary somatosensory area of the brain is located very close to the primary motor area of the brain, because our movement is largely linked to what we experienced before. For instance, we detect the pressure required for punching keys of a keyboard, the immediate feedback of a pencil scratching on paper, and therefore we can adjust our motor output in respect of these. Just below the lateral fissure, the primary auditory area sits. It is located on the transverse temporal region of the gyrus known as Heschel's gyrus, whilst the occipital pole and the area around the calcarine sulcus is known as the striate cortex. This is where the primary visual cortex is located. Let's summarise the primary projection areas that we have seen so far. The primary motor areas are where motor descending pathways originate. This is known as the primary motor cortex and is found on the prefrontal gyrus. The sensory pathways each have a different function and location depending on where the sensory pathways are being stimulated, and they each terminate in a specific region. The general sensory, sometimes called somatosensory or somesthetic, is located on the postcentral gyrus, whilst the visual is located in the striate cortex, which is formed by the occipital pole and around the calcarine sulcus. The primary auditory area is found in the temporal region on a gyrus known as Heschel's gyrus. So that's the primary projection areas, but what about the secondary or association areas? Well, it would make sense that the association areas are located near to their primary areas, so that's what we find. The motor association areas are located near to the primary motor area. The premotor cortex role is to finely tune movements, such as for spatial guidance. The supplementary motor area is thought to control our trunk muscles in response to posture. The somatosensory association areas is located near the primary somatosensory area. Its role, as with many of the other association areas, is not as well understood, but it is believed to receive a lot of sensory fibres from the thalamus, suggesting that it is connected with the memory of a kinesthetic touch. For instance, if we close our eyes, we can interpret the difference between two similarly textured objects, such as a feather and cotton wool. The general somatosensory association is likely responsible for being able to identify different general touch based on its experience with a previous encounter. The auditory association area is once again located near the primary auditory area, in the temporal lobe. It is responsible for processing acoustic sound signals that the brain interprets as sound, speech, or music. It forms a part of the brain known as Wernickes area, which we will come on to in a few minutes. The visual association area is found in the pre-striate cortex, around the primary visual cortex. This area is responsible for interpreting shapes and orientation. We also have a second visual association area, up by the premotor cortex. This is called the frontal eye field. This area is responsible for movements of the eye, both voluntary and a movement called saccadic eye movement. Saccadic eye movement happens in response to stimulus in the visual field. For instance, if you were to sit perfectly still in a room and a rat scurried across your peripheral vision, your eyes turn towards it in a reflex action. We are drawn to movement. Let's summarise the association areas. The secondary motor or motor association areas send output to the primary motor area and organise patterns of movement. The premotor cortex is anterior to the precentral sulcus on the lateral surface, whilst the supplementary motor area is anterior to the precentral sulcus on the medial surface. The frontal eye field is anterior to the premotor cortex. The secondary sensory areas receive input from primary sensory areas and are involved in interpreting and understanding information. General sensory areas are in the superior parietal lobe. The visual association areas are in the pre-striate cortex, and the auditory association areas are located within the superior temporal gyrus around the lateral sulcus. Let's go and have a look at the next learning objectives for this lesson. and use the opportunity to have a little breather from the huge amount of information I've just passed over. So we've looked at the first 2 learning objectives, and now let's go and cover the final 2 learning objectives, which will hopefully enable you to see the bigger picture. So the prefrontal cortex is the cortex essentially right at the most rostral region of your frontal lobe. We already know the frontal lobe is responsible for a lot of motor-related functions, but let's have a look at areas in front of the designated motor projection areas. So the prefrontal cortex, predictably, is associated with motor function, but in addition to this, it is also responsible for regulating emotions, which is where I will introduce you to Phineas Gage. Phineas Gage was a railroad worker, and whilst on the tracks, he got himself into a little pickle. Unfortunately, there was premature detonation of dynamite, and one of the metal rods that Phineas was working with shot up and pierced through his maxilla, travelling through his orbit and out of the top of his head just behind his forehead. Thankfully and somewhat miraculously, he survived, losing only his eye and with severe damage to his frontal lobe. He was well in general terms, but damage to his frontal lobe resulted in him being impulsive. He lost the ability to form higher order judgment and inhibition that we form in order to live in society. Unfortunately, it wasn't that he just didn't care about his behaviour. He wasn't being naughty. Instead, the reason and understanding of acceptable social behaviours just wasn't there anymore. Similar symptoms can be seen in patients with certain frontodementias or after head injuries. It's not a conscious choice that the patient is making. They genuinely don't understand why their behaviours are unsatisfactory or antisocial. The frontal lobe we know is the location of the primary and secondary motor areas. It is home to the precentral gyrus. It is the origin of motor efferent fibres. But what hasn't been made clear is that these motor fibres affect the contralateral side of the body. That means the left hemisphere precentral gyrus controls the movement of the right side of the body, because the fibres descended through the brainstem into the spinal cord cross over at the medulla. The reasons why is not something you need to worry about at this stage in your neuroanatomy journey, but it is suffice to understand that the corticospinal tracts make up about 90% of the efferent fibres found within the precentral gyrus. After much research, neuroscientists and anatomists have been able to approximate the loci of each region on the precentral gyrus in a map or in a schematic called the motor homunculus. We could see that the general motor function for the feet and the leg are found within the medial cortex of the precentral gyrus. Whilst we travel more laterally, we could see that the arm, hand, face and eventually tongue, pharynx and larynx fall along this gyrus. You'll notice that some of the body parts are larger than others. This is because we have proportioned out the dedicated number of fibres to each region. When we look at the face and fingers, we can see a large proportion of fibres are dedicated to movements in these areas. When we think about the precise and frequency of movements made in the hands and face, it is no wonder that a huge number of neurons are allocated to these parts of the body. We make multiple facial expressions a day, all day, every day, and the fine motor skills of our hands have been necessary for our survival. The parietal lobe we have already discovered houses the postcentral gyrus, so we know it is responsible for general somatosensory perception. Again, this is contralateral, so the right hemisphere postcentral gyrus receives messages from the left side of our body. Again, after gentle probing and mapping out, we have been able to provide a sensory homunculus. and you'll notice that this pattern is very similar. A huge proportion of the sensory neurons are dedicated to our face and fingers because it is hugely important that we are able to determine things that are safe and things that are not safe to be in the regions of these vital pieces of anatomy. The occipital lobe and the frontal eye field in the frontal lobe are our visual projection areas. And if we look at this table, we can see a summary of the roles of these areas, which we've already covered in this video. Pause the video if you wish to recap these, but for now I'm going to move on and discuss the auditory and linguistic functions of the brain. There are two linguistic regions to touch on. One of them is our ability to produce language, which is this area here, called Brocas area. Given that it is responsible for the production of spoken word, it makes sense that it is located near the motor regions of the brain. The other region that we have previously mentioned is Wernickes area, which is located around the auditory association area of the temporal and some parietal lobe. Brocas area is on the anterior precentral gyrus and functions to generate speech, whilst Wernickes area is for the comprehension of written and spoken language. What we know about linguistic function is that the left hemisphere of the brain is normally dominant. We briefly introduced this lateralisation of function. In about 90% of the population, the left hemisphere will be the hemisphere that controls and processes language, in both written and spoken form. If the left hemisphere is damaged in Brocas or Wernickes area, we can expect that there will be significant damage to language production and or comprehension. This is called aphasia, and there are two types of aphasia that perfectly demonstrate the outcomes of the damage. In the final few minutes of this video, I'm going to share two clips of patients with aphasia. The first clip is a result of damage to Brocas area, resulting in Brocas aphasia.
So Clara, how did you get your injury?
Home. Dizzy. Lightheaded. Fall. Friend. Call 911. Ambulance, doctor, stroke, right, side, left, hemisphere,
How has your recovery process been?
slow, difficult, remembering, expressing.
You'll notice that the words make sense, but that the patient really struggles to form those words. They are stuttered and sporadic. The motor generation of words is clearly difficult, even though it is clear that she knows what she is trying to say. You'll also notice that this patient is describing her traumatic brain injury. She describes how she had a left hemisphere stroke. She describes how this has affected the right side of her body and resulted in Brocas aphasia. As terribly unfortunate as this is, it is an almost textbook example to summarise the topic of today's video. In contrast, This second clip demonstrates Wernickes aphasia.
Hi Byron, how are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment and talked with the people for them over there. They're diving for them at the moment. They'll save in the moment. He'll have water very soon for him. With luck for him.
So we're on a cruise and we're about to
We
get to
will
Juneau.
store it right here and they'll save their hands right there for them.
And what were we just doing with the iPad?
Right at the moment they don't show a darn thing.
With the iPad that we were doing. Like here?
I'd like my change for me and change hands for me. It would happen. I would talk with Donna sometimes. We're out with them. Other people are working with them. I'm very happy with them.
Good.
This girl was very good and happy. And I play golf and hit other trees. We play out with the hands. We save a lot of hands on hold for us.
This gentleman is speaking very fluently, but the words he uses don't make a great deal of sense. He also doesn't seem to realise that his sentences, although beautifully phonated, aren't transmitting the information in a manner that we can understand. And that concludes this video. We have looked at the brainstem and the cranial nerves, and the primary and association motor and sensory areas, and located them on an adult brain. We have rounded it off nicely, with taking a minute to look at the two different types of aphasia.